Thank you very much. My name is Anthony Gitter. I'm currently a postdoc at MIT and Microsoft Research, but I'll be talking about my PhD work at Carnegie Mellon with my advisor, Ziv Bar-Joseph. And the goal was to reconstruct disease-relevant signaling pathways de novo. So the running example that I'll use throughout the entire talk is the response to viral infection. Many of you who were in the afternoon session in this very room yesterday will know that there is a lot of interest in detecting protein-protein interactions between pathogen proteins and host, in this case, human proteins. So the question I'd like to answer is, well, once we know that the human cell has some way to detect the pathogen, what does it do with that information? The answer is something very complex that we can't observe very well. But what we can observe are some downstream changes, such as temporal changes in gene expression. That's not very satisfying on its own. What we'd really like to do is explain some potential mechanistic methods that link these upstream proteins, receptors, or human proteins that interact directly with the pathogen to the downstream changes in expression. And we know that there are DNA binding proteins like transcription factors that are driving some of the temporal expression changes, and that hidden within very complex and dense human protein-protein interaction networks are some signaling pathways that connect upstream sources to these transcription factors and pass along the information that there's a virus in our environment and we need to respond to it and initiate these transcriptional changes. So to be a bit more precise about the computational problem, I'll take a human protein-protein interaction network, some approximation of where human transcription factors are binding DNA, and both these types of data can actually be condition independent. I don't need to collect protein-protein interactions, say, in the specific context of a viral response or when the virus is present in the cells. I do need some condition-specific data, namely temporal gene expression data, and some idea of what source proteins are detecting the pathogen or the virus and initiating this response. The algorithm that we've designed to deal with all these data and produce signaling pathways is called SDREM, the Signaling and Dynamic Regulatory Events Miner. Now, I'll only briefly overview this algorithm. It's been developed in many phases over many, many years. The first part looks at the transcription factor binding and the gene expression changes and tries to figure out from the temporal information in the gene expression data which transcription factors are driving these transcriptional responses. And this was described by my colleague Jason Ernst many years ago. Then once we have some candidate transcription factors, how are they connected to the source proteins? And what are the signaling pathways and signaling cascades that might connect the upstream sources to these transcription factors? This is work we presented in nucleic acids research. And lastly, how do we unite these two phases? So we developed an iterative algorithm. This is the full SDREM algorithm that passes information back and forth between these two sides. So we want to find transcription factors that make sense in both contexts. They explain the gene expression data well, but they're also connected to the sources, because if we have some transcription factor that doesn't seem to have the capability to receive any messages from the source proteins, it doesn't really seem likely that it's going to be a key participant in the immune response to, say, a viral infection. And this is the SDREM algorithm in total, which we described earlier this year in genome research. But that's not actually the focus of my talk today. Today I want to talk about how to make SDREM more translationally relevant. How can we make SDREM relevant to, say, potential therapies? And the way we do that is by incorporating new types of functional data that allow us to find more condition-specific and disease-relevant pathways, and also by using SDREM's predictions to produce very specific types of predictions about what the effects of node removal would be in these signaling networks. I need to dive into part of the SDREM algorithm in a bit more detail to make the later parts of these extensions somewhat more comprehensible. So I'll talk about how we find directed signaling cascades. So we have these source proteins, which could be, again, as I said, proteins that interact with a pathogen or, say, particular innate immune response receptors. And we know that these proteins are initiating some message and that information is flowing through a network. But the information may flow in perhaps conflicting directions, and the goal is to find a single orientation that's relevant to this condition or disease. And the way we do that is by actually enumerating all very short source-target pairs to identify potential candidate pathways that connect the sources and the targets. And for each candidate pathway, we can give some score to this pathway, a weight on the pathway that's the product of elements along the pathway. So these are the confidence in the individual interactions, as well as some weight on the transcription factor target that came from our analysis of the gene expression data. So here we have pathway 1 with some associated weight. We can enumerate some other pathway 2 with a different weight. And the question to ask then is, how should we orient each edge with respect to these pathways and their weights? So if I orient the edge from right to left, I'll see that I can still pass information along pathway 1, but pathway 2 has been broken, so it should no longer count towards some global score. If I flip the edge direction the other way, I now get a score for pathway 2, but not pathway 1. So you can get some intuition about how this sets up some global combinatorial optimization problem, where my objective is to pick an orientation on all these edges that's going to give me some set of paths that still allow information to flow from the upstream proteins that detect the disease to the downstream transcription factors that are driving the transcriptional response to the disease. So moving on to the new extensions, and specifically the new types of data we'll incorporate are RNA interference screens. And the challenge, just to remind you, was to help scale for model organisms like yeast, which have relatively few proteins, depending on if you think 6,000 or so is a large or small number, to much larger human interaction networks that are more dense and have many more interactions. And the problem is that in human interaction networks, there are many equally good paths that could connect a source and target, and that makes the edge orientation problem quite challenging. What we'd like to be able to say are which nodes in this network might be more relevant to the disease or to the context we're studying, so that we can give preference to those nodes in the pathways. So RNAi screens, to give a very brief introduction, give some type of functional association between a gene and a phenotype. So one phenotype might be cell viability or viral load. And we can see that in wild type, we have some measurement for this phenotype. And when we knock down a gene, we'll perhaps get some different phenotypic value, like increased viral load or decreased viral load. And that allows us to associate the knockdown gene with the phenotype or the viral infection or whatever our condition of interest is. The problem is we can't just take these RNAi screen hits directly and constrain our paths to use them and them alone, because RNAi data, while very rich and providing a lot of information, is incredibly noisy. And as we've seen in genome-wide studies for H1N1 influenza and for HIV, these results are just not reproducible across labs. So in both of these cases, we have something like 1,000 hits, more or less, for each virus. Yet the number of genes that are detected in all of the genome-wide screens for a particular virus is 3 to 7. So there's clearly a lot of off-target effects, which is a well-known phenomenon in RNAi signaling. And our goal is to use SDREM and some pathway context to help explain the RNAi data better. So what we can do is transform the data we get from an RNAi screen into some vertex weight. And we can see that it's very easy to incorporate vertex weights into the pathway scores. Now we've just extended the definition of a pathway weight to include terms on the vertices. And if a node is not implicated in an RNAi screen, we can just give it some default background weight. And now the end result of this is that we can prefer pathways that go through many genes that are already functionally associated with the disease of interest. And we can also look at other screen hits in the RNAi screens, such as this highlighted node on the right, that don't really make a lot of sense in the context of the pathways, and we can filter them out or disregard them and perhaps suggest that there's some off-target effect or some product of noise. Now the other question is actually a bit more interesting, is given an SDREM model, how can we predict the effects of an RNAi screen that hasn't been performed yet? And if we can do this well for many viruses and diseases, I think it would have great value in suggesting which genes we should be looking at for perhaps wet lab follow-up or clinical follow-up. The key point that lets us do that is that SDREM produces directed signaling cascades. So if I remove some node in this network, like the highlighted node, there's a very clear sense of what's downstream of that node. In this case, we see that there's a single transcription factor downstream of the node. So if I pull this node out of the network and sever its paths, this transcription factor no longer has the capability to respond to the virus. It is not being informed that the virus is present. In other cases, a node removal may have some smaller effect on many transcription factors. In other cases, it may have a very marginal effect on a single transcription factor. And hopefully this is giving some intuition that removing different nodes from the network will have varying levels of effects. And we can actually use those predicted effects in the network to try to estimate what will happen in a potential RNAi screen. And I'll just give intuition about the metric we developed to score these things. There's a score that tells us the fraction of transcription factor connectivity that remains after we perturb the network by removing a node. So the details are, of course, in our paper, but this should help give some sense of what we're scoring and what we're doing here. But given the directed pathways and given such a score, we don't need to limit ourselves to single nodes either. We can actually ask what happens when we would hypothetically knock down pairs of nodes. So here we see redundant paths in the predicted signaling pathways. So removing a single node won't have a very substantial effect. But a pairwise knockdown would be predicted to have a very strong effect because, once again, we're completely cutting off all paths to this transcription factor. And we can actually look to decades-old studies in yeast that tried to quantify genetic interactions experimentally. And the way that they assessed epistatic relationships was by looking at the expected effect of a double knockdown or a double knockout in yeast and the observed effect of the double knockdown. And the expected effect decomposes into the product of two observed effects for single node knockdowns. So in yeast, these would be something like colony size after doing a knockout. In our case, they're going to be this in silico observed phenotype. So we have the same score from before that tells us what fraction of paths survive the knockout to transcription factors. And we can calculate this, again, for single nodes or for pairs of nodes or even for higher order combinations of nodes. And this gives us a way to calculate all of the terms in the above equation, the in silico observed phenotypes for each single node and pairs of nodes. So let's see how this does by studying influenza infection. We studied two strains of influenza. H1N1 is actually a seasonal strain of H1N1 that we studied. It's been very well studied, a lot of papers targeting this. And on the other hand, we have H5N1, which is the bird flu or avian flu. This is a highly pathogenic strain. The human system clearly responds to it very differently. And we have specific types of data for each virus. So the sources are, again, human proteins that interact with the viral proteins, plus some short list of innate immune receptors. We have, of course, more for H1N1, which I said has been studied more thoroughly. We also have the RNAi screen hits. For H1N1, these have been performed genome-wide from different labs. For H5N1, there's very little that's known. We only have a single targeted screen, which produced just 32 hits. And we have temporal gene expression data for both. I'd like to stress just once more that H5N1 is highly pathogenic. It's actually difficult for labs to work with. In that case, we have less data available, and we also have more value for any successful predictions because virologists could really benefit from a short list of single gene predictions or pairs of genes predictions that they should target in a lab for functional studies. And here's what the SDREM models look like. So we built an SDREM model for H1N1 influenza response, and this shows the temporal transcription factor activity as derived from temporal changes in gene expression. So along the x-axis, we have time. Along the y-axis, we have log2 fold change. And these big groups, the thick bars, are something like clusters of genes that respond similarly over time to the influenza infection. And what we're really interested in are the points where these groups diverge. So genes are expressed similarly for a certain amount of time, and then some transcription factor or groups of transcription factors come in and exert their regulatory force and cause a split because they bind a subset of the genes, and that subset does something different now. For example, we have this green bar that diverges at 4 hours, and SDREM has annotated all these divergence points with the transcription factors that are likely controlling these changes. If we look at these transcription factors, we find most of the major immune response TFs, we have IRFs, we have NF-kappa B, et cetera. And the other half of the SDREM model are the signaling pathways. So how do we connect these red source proteins that we were given as input to the green transcription factor targets that are just the same transcription factors that I showed you on the last slide? And there's also blue internal signaling proteins that are sitting between sources and targets on the pathways. I'm also showing in diamonds the genes that were hits in one of the RNAi screens. And there are also some other circle nodes that are predicted to be relevant to the disease, but didn't show up as hits in any of the screens, perhaps due to redundant pathways. So the predictions we make are enriched for very relevant GO terms. We also do a good job of finding pathways that go through many of the RNAi hits, which is what we'd like. Some of you may find this figure to be a bit dissatisfying because it doesn't really convey the sense of specific pathway predictions. It looks like a hairball, which is a word with very negative connotation in this community. The good thing is I am providing very specific pathway predictions. So if I focus on a single gene like HIPK2 in this network and only show the top path through this single gene and then rearrange the edges, you see a much more clear, beautiful picture of how this gene is functioning in the immune response. So there are certain sources that are upstream of it, there are certain transcription factors that are downstream, and we can see what would happen if we were to remove this node from the network. Now, how do I know which nodes to visualize? Well, I have the ability to provide a ranked list of all genes in the model based on their expected knockdown effects. So I can ask for all genes, what would happen if I knock this down? How would transcriptional activity be affected? And I can use that to sort my genes and look at specific pathways through these genes and again, find very specific mechanistic explanations for changes in transcription factor activity. So I still have to convince you that we do a good job of predicting these RNAi effects. Thankfully, we do. What we can do is run SDREM again using only the temporal gene expression data and the sources and hold out all of the screen data for H1N1 and then just use the directed pathways to predict effects. And what we find is that when we rank all the genes by their expected effect, we see that at various thresholds we have significant overlap between our predictions and the H1N1 screens. This is especially impressive considering that the H1N1 screens don't even agree amongst themselves. So using this same strategy, we can now ask, well, what happens in H5N1? Why is the host response to H5N1 so different? Why is it so much more deadly? And we can actually find within our top ranked genes some predictions that appear to be exclusively relevant to H5N1. So these would be great candidates for experimental follow-up in a lab because they're not H1N1 hits, they're not directly interacting with the virus, so that there was previously nothing to suggest that they were relevant to this type of virus. And moving on to the double knockdown predictions, which I think are actually the most appealing part of this work, because right now there's just no way to systematically screen pairs of genes in mammalian cells. It's been done for yeast. Yeast has fewer genes, but you can't take all 20,000 by 20,000 genes and just run through this list of millions of pairs using current experimental methodologies. So what SDREM can provide is a short list of, say, 100 pairs, 1,000 pairs, whatever you have a budget to test, and help guide you to very interesting epistatic relationships between pairs of genes that would be very, very difficult to find otherwise because they have, by definition, surprising relationships to the viral infection. And I'm just highlighting a couple from our top-ranked pairs right now. This particular pair is interesting because it has low degree in the network. So if you remove a low-degree protein, you would expect that it doesn't affect many downstream transcription factors. But here we see that removing this particular pair actually happens to affect many transcription factors, even though neither node removal in isolation has as substantial an effect. So to wrap up, we have a very general algorithm that we've applied in many other species and conditions besides human disease called SDREM. We've validated it in yeast, studying the osmotic stress response, and it gives directed signaling pathways and temporal predictions about transcription factor activity. We have code available. We've extended it to make it more relevant to human disease by incorporating new functional data. But really the key point is that we can go from limited condition-specific data, say the condition being viral infection. We only know the transcriptional changes in the upstream source proteins. From that, we can give you directed pathways, and we can use the directed pathways to make accurate predictions about perturbing those pathways. And even more so, we can attempt to make pairwise knockdown predictions that give very interesting pairwise candidates to test in a lab. And this simply couldn't be accomplished by screening millions of potential pairs just blindly. And it also gives us a way to do targeted experimental work for pathogens like SARS and anthrax and avian flu that are actually quite difficult to work with experimentally. So I've had many great collaborators on this work, especially my advisor, Ziv Bar-Joseph, others at CMU and Pitt, experimentalists at the Whiteman Institute, led by Nama Barkai. And I'd also like to just give general thanks to everybody in the Bar-Joseph lab and now the Frankel lab at MIT, one of my postdoc advisors, and my colleagues at Microsoft Research. And again, we have code available. This is an easy-to-use system, and I'd be happy to help if anybody has specific experimental questions or diseases they'd like to study. Thank you very much.
Okay, we have time for plenty of questions.
How do you get the weights for your network?
The protein interaction weights.
Yeah.
Right, so I showed an equation with these WEs, where E's are edges. This is in the supplement of our bioinformatics proceeding papers. We've worked with experimentalists who study protein-protein interactions, and they've helped us place a confidence on different experimental platforms, say yeast to hybrid related things, mass spec related things, crystallography, and then we can use the number of independent experiments that detect an interaction and the type of experimental methodology that was used in all those experiments, and combine that into some aggregated weights on all the interactions. It's a general philosophy that's being used by a number of other labs as well. I can go into it in a bit more detail later. Yes?
I'm curious why the directionality is so important to you. I understand algorithmically how it helps, but you would think those interaction networks are bidirectional, and imposing a strict directionality on them is non-biologic.
So the motivation there comes from what we see in signaling pathway databases. So if you go to KEGG or Reactome or BioCarta, you see directed signaling cascades because many of the members along this cascade are kinase substrate pairs where we have specific post-translational modifications that don't work in both directions. They really are directed. But some of the experimental methodologies used to tell us that these pairs are doing something interesting don't provide the directionality. So this is what gives us an undirected protein-protein interaction network, and we'd like to have some directed signaling cascade. The caveat is that when we direct members of a complex, this is a big approximation. We do that for very technical reasons that I don't have time to explain, but it's come up again and again in response to reviews, and there's a justification there that's